To start EO6, we're going to talk about bacteria. As we learned last semester, the three domains of life are bacteria, archaea, and eukarya. Bacteria and archaea, if you recall, are prokaryotic organisms, meaning they don't have a nucleus. And so this lecture is going to focus on bacteria primarily, a eukaryotic organism that is microscopic, we cannot see with our own eyes. On the right-hand side of your paper, for your notes, please write that bacteria are prokaryotic, which means they don't have a nucleus, and that they are single-celled. So every single bacteria is going to be only one cell. The whole entire organism is a cell. As you can see here, they are measured in micrometers, and a micrometer is 1/1,000,000th of a meter. For example, if you took a meter stick and you looked at all the little millimeters on it, that's 1,000th of a meter. If you broke that millimeter into 1,000 smaller pieces, that would be 1 micrometer. And as you can see here on this image of Escherichia coli, that bacterium is 1 or 2 micrometers in length. So about 2 micrometers or 2/1,000,000ths of a meter in size. Jumping back to the left-hand side of your paper, you can see this image on your sheet. And the main thing I want you to understand with this is that when we talk about bacteria, it's an organism or a group of organisms that we don't typically think a lot about, except when it comes time for us to be sick or that kind of thing. But if you look at this, bacteria are a huge portion of the life that exists on Earth. Of the three domains, bacteria, eukaryote, and archaea, you can see here that bacteria is a massive, massive portion of the tree of life, that it is huge. It contains the most species in existence. And so when we talk about bacteria, when we talk about life in general, we often think about humans and animals as our default, but they are such a minor portion. If you look at this little branch here, that's eukaryotes. This teeny little portion are all the eukaryotes that exist on Earth that we're aware of. So comparatively, they are not similar in size at all. Bacteria are typically found in 3 shapes. The shape on the left is a spherical shape, and we call that coccus. The way that I can best remember coccus is the sphere is that it has the C's and the O's, which are more spherical in shape. So we can see here, coccus are the sphere, kind of round bacteria. Bacillus are these rod-shaped ones. As you can see in the image here, we have more of a rod, kind of like a pill shape. And the way that I remember that is the 2 L's in bacillus are kind of like those rods. And then our third and final shape is Spiralia. And as you can see here, Spiralia is like a spiral, kind of has it in the name, right? So we have that spirally shape on the right. For the structures of bacteria, you don't need to know every single one of these, but I would encourage you to write them down and copy them to your notes. The couple of main ones that you want to know are the cytoplasm, because every cell has a cytoplasm, no matter if it's prokaryotic or eukaryotic, the DNA or the plasmid, along with the nucleoid, because every living thing has genetic material, the cell membrane, because every living thing has a cell membrane, and then the ribosomes, because every living thing has ribosomes. One other important characteristic of bacteria is the cell wall, which is made out of peptidoglycan, and this helps us identify which type of bacteria we're looking at when it comes to diagnosing illness. Another key piece of a bacterial cell is the flagellum, and you can see that here is at the end of this bacterial cell to help with movement. All right, so flagella are going to help with movement. These pili, they're going to help that bacterial cell stick to the cells that it's going to infect or be a part of. So those are different structures that you will want to know. One important characteristic about bacteria is that they can either be heterotrophic or autotrophic. And if you recall from last semester, autotrophic meant that the organism makes its own food. Autotroph means self-nutrients. And so this organism can make their own food from an energy source. They can either be photoautotrophic or chemoautotrophic. Photoautotrophs are going to get their energy from the sun. We learned about this with plants. But bacteria can also be chemoautotrophic, meaning they can get their energy from inorganic sources. So non-living sources can provide them with energy. Heterotrophs, on the other hand, are organisms that cannot make their own food. They need a different nutrient source. That's what heterotrophic stands for. And some bacteria are going to need to consume other organisms to get their nutrients. The key takeaway that I would like you to take from this slide is the reproduction aspect of bacteria. Bacteria are prokaryotes, and they're going to reproduce asexually, meaning there's only one parent. One parent divides and becomes now two new organisms. They do so through a process called binary fission. Binary fission, I'm going to spell out for you here. There we go. Binary fission is the process by which they divide. It's very similar to mitosis, but it is not mitosis because they're prokaryotes. They don't have a nucleus. Mitosis is division of the nucleus, and therefore it cannot be called mitosis because it doesn't have a nucleus to divide. So instead we call it binary fission. Binary means two, so from one we're getting two, and fission means like a separation. So they are, one cell is separating to become two brand new cells. One awesome feature about bacteria is that they can exchange DNA with other bacteria. That would be like if I came in contact with another human being, or just by coming in contact with their DNA, I could pick it up and incorporate it into my own DNA. Bacteria are able to do that, and they do so through two ways. Conjugation, which is when they directly exchange it from one bacteria to another. So they actually come in contact with each other through a little filament, and they transfer that DNA from one to the other. Transformation is another way that they can exchange that DNA by if another bacterium bursts and there's fragments of their DNA around, or they just come upon other DNA, that they can just absorb that into their that cell and incorporate that DNA into their DNA. This is one important factor about bacteria that makes them able to evolve very rapidly, and is one of the biggest struggles that we face when it comes to creating antibiotics that are effective against them. They are able to change so frequently and take on other DNA components that could be beneficial that it makes it hard for us to continually work to take them out. The last thing I want to look at here is just a couple diseases that are caused by bacteria. Not all diseases are going to be caused by bacteria. Some are viral, some are protal, and some are fungal. And so a couple key ones here that are caused by bacteria are pneumonia, which is, it can be bacterial or viral. Staph infections, methicillin-resistant staph infection is going to be a very big illness that we'll talk about this unit. Tuberculosis, typhoid fever, food poisoning, gonorrhea. You can see all of these here are going to be anthrax, Those are important diseases that can be caused by bacteria. And you can see a couple others that are listed on your notes. You don't have to write anything there, just kind of read through that. Look at what the disease is called, the specific bacteria that is causing it. So that's its scientific name, its genus and species. The area that it affects in the body, like botulism, is caused by Clostridium botulinum, and it affects your nerves, your nervous system. And then the mode of transmission, so how it gets into us as humans, is when we eat improperly preserved foods. So typically you find botulism outbreaks happening with like improperly canned foods and things like that. But that is all we have right now for bacteria. We'll continue to work more with this unit.